Let me remove the face mask. Our life has become different now. Vanakkam. I'm Ammasi Periasamy, as many of you know, and trying to, thanks, I wish I should be in person, but because of the situation, I am sending this video. So thanks for Professor Hamalada asking me to participate in this international conference. And thanks for all the organizers involved in the conference. According to CDC, Center for Disease Control in USA, between 2007 and 2020, the number of deaths is expected about 15% for men and 8% in women. So the rate of cancer deaths is about 100,000 people in the United States, expected to keep going down. So as you see in the graph, the diagnosis increases about expected increase in the diagnosis is about 630,000 in 2020, but the death rate decreases. So these are all due to the improvement in technology advancement. On the top of the list, you can see the prostate cancer. So I am going to talk about the prostate cancer, how we developed a technique to detect the prostate cancer in early stage. So prostate cancer is a treatment and diagnosis of prostate cancer is a much debated subject. Prostate specific antigen, whenever you go to the hospital, they will ask you to take a PSA test particularly for men. Needle biopsies also used for different parts of the tumor to test the prostate cancer. So we are using endogenous molecules to investigate the prostate cancer cell response for drug treatment. So the technique we use to detect the prostate cancer is quantitative FLIM, fluorescence lifetime imaging, and data analysis. And then we monitor the optical redox ratio, so which will tell you the metabolism of the condition of the cancer. So the terminology I am going to use in my presentation is mainly the NADH, the coenzymes, NADH bound and free is investigated in the mitochondria, and the FAD, flavin, and amino acid tryptophan. So these are the coenzymes and amino acid I will be investigating in prostate cancer. We use the prostate cancer cells, and also I am going to talk about the leukemia cancer, where we use acute myeloid leukemia cell, AML cells. The drug to test the prostate cancer, we use doxorubicin. And for the leukemia cancer, we use the drug ceramide nanoliposomes. The technique we use the multi-photon fluorescence lifetime imaging microscopy, and also we developed the technique called fluorescence lifetime redox ratio. The data analysis approach, which is a FLIM-FLIRR, and also we introduced a machine learning because a huge number of data is available. We wanted to analyze the data as fast as possible. We are very much experienced in developing, training the students and international scientists on lifetime imaging and FLIRR, and we are investigating on the research part, as I mentioned, endogenous molecular imaging. I am now the pioneer, developed a fluorescence lifetime imaging in 1990s in the University of North Carolina, Chapel Hill. At that time, there is no imaging device. Now the imaging is available, the whole electronics made into a single PC, you can buy from number of commercial lifetime imaging systems. So as I mentioned, I will show you some data about cancer metabolism. The recent evidence has suggested that cancer should be considered as a metabolic disease. Growing tumors are rewired with the metabolic programs to meet and even exceed the bioenergetic, biosynthetic demands of continuous cell growth. The metabolic profile observed in cancer cells often includes increased consumption of glucose and glutamine, increased glycolysis, changes in the use of metabolic enzyme isoforms, and increased secretion of lactate. Recent research in the field of cancer metabolism raising few questions. Why do cancer cells shift their metabolism? Are the changes in metabolism in cancer cells a consequence of the change in proliferation or a driver of cancer progression? Can cancer metabolism targeted to benefit patients? There are a number of techniques are used to measure the cellular metabolism. So I will be using fluorescence microscopy techniques, which is fluorescence lifetime imaging of the NADH, of the coenzymes in mitochondria. So what is lifetime imaging? Lifetime measurements means it's a biophysics. When you excite the fluorescent molecule, it goes to the excited state and return to the ground state. During that time, it releases the photons. There are a number of reaction takes place during this decay or in the Zablonsky diagram or in this process. The occurrence of fluorescence is in the nanosecond process. Many events of cellular process can be measured spatially and temporally. Compared to any confocal microscopy measurement, the lifetime microscopy is excuse me, insensitive to environmental changes of the biological process, insensitive to change in molecular concentration or increase in fluorophore, or insensitive to power of the excitation intensity. So lifetime technique provides you the quantitative measurements of the signal compared to the intensity-based measurements. I also edited a book on fluorescence lifetime microscopy. It is a beginner and for advanced level people. And also we wrote a review article in the Nature Protocols, plus and minuses of the lifetime imaging techniques. Cellular energy metabolism with a focus on NADH, that's what we are working on. Glucose degraded to pyruvate upon reduction of NAD plus to NADH in the cytoplasm. Mitochondria is in the cytoplasm. Pyruvate is preferably converted to acetylcholine and fed into the TCA cycle in the mitochondrial matrix. The proton gradient built over the inner mitochondrial membrane is used by complex 5 to convert ADP to ATP. So there are a number of articles published in the Cytometry A special issue during January 2019 issue. You should, if you are interested, you can go through a number of approaches, how to measure the metabolism in cells and tissue. So here is an illustration, imaging of, this is the green color represents the NADH signal, which is in green in the cytoplasmic area. Mitochondria is labeled with mitotracker fluorescent probe. And if you co-localize, you can see whatever the NADH signal we are measuring is coming from the mitochondria. So the technique which we developed is called FLIRR, fluorescence lifetime redox ratio, which this provides very sensitive way of measurements, not only in cells and also in live animal. The fluorescence lifetime is a powerful technique for this NADH to measure the autofluorescence of NADH in FAD and tryptophan. This provides autofluorescence. You do not have to label the NADH, FAD, and tryptophan. You excite with a wavelength, it provides a particular wavelength emission for NADH and FAD and tryptophan. Potentially, FLIM assay may be more versatile and predictive method. It has been reported in the literature by biochemical methods that the degradation or reduction of tryptophan level occurs in cancer. We also use the FLIR method would allow us to investigate the mechanism of degradation of tryptophan. Combining FLIR and FLIM may provide detailed information of cancer progression. So upon treatment, cancer cell-specific metabolic reprogramming occurs. Depending on specific treatments and cancer type, metabolic changes occur. Interaction of oxidative phosphorylation and glycolysis is affected. Tryptophan levels are affected in cancer. Change before and after treatment. Heterogeneous cellular responses to treatment are observed. So conventionally, the redox ratio is calculated. Redox ratio is a measure of metabolism. Is calculated using intensity-based method, which is ratio of the fluorescence of NADH and fluorescence of FAD. How the lifetime measurements can be used to calculate the redox ratio. So in the lifetime technique, what we are using is ratioing the bound coenzymes of the NADH divided by the bound FAD. So this provides avoids all the artifacts involved in the metabolism measurement, cellular metabolism measurement. If you know how to process the data lifetime, this is how the bound coenzymes of NADH can be extracted from the lifetime data. So how do we go about it? We tested with the cells, and then we did with the animal, live animal, and we found out that the data using intensity-based method, we found out that data was not reproducible. Here it is we are using 740 nanometer in the animal. The dye was injected in the ear of the animal. And here's the images, which shows the lifetime images of NADH. And this is the lifetime image of the FAD. This is the autofluorescence image. And this is the selected area of the animal, ear animal. So here you see the intensity-based redox ratio. It jumps all over the place. It is not even. It's supposed to be even, as shown in our method. It may be due to the differential absorption and scattering of light in the tissue, and due to different excitation intensity used in varying depths. So because of the absorption, light scattering provides a lot of artifacts using the intensity-based redox ratio measurement. When you use the lifetime-based, the method we developed, it is almost uniform. There is no variation in signal in depth or in any other way of measurements. So we developed the FLIR method is kind of region of interest based single cell analysis. And as you know, the distribution of the signal is heterogeneous. That can be monitored by using the region of interest in single cells or in a live animal. So we compared the intensity-based method versus our way of doing the lifetime technique. So prostate cancer cell, we use the imaging system here as I shown here, and you have the tryptophan PMT, NADH lifetime measurement, and the FAD measurement. Simultaneously, you can measure 3 signals at the excitation wavelengths. As you see, the tryptophan emission is in the UV. Most of the microscopy emission optics is in the visible spectrum, 400 to 700. But this microscope is designed to collect tryptophan 300 to 400, which is the UV spectrum. So you have to adjust our optics to measure. The NADH emission is 400 to 440 to 480. The FAD emission is 520 to 560. The cells we are using for prostate cancer cell is LINCAP, Caucasian prostate cancer. And also we used African American prostate cancer cell line. We used the toxorubicin as a drug and collected the images 15 minutes, 30 minutes, and 45 minutes and 60 minutes images. And we processed all the data according to our software. You feed the images, the software spits out the data. So how do we, our region of interest software works? As you know, the mitochondria is in the cytoplasm, and we created the software. It automatically goes and picks out the mitochondria area in the cytoplasm. This also helps, not only you can measure the rate of metabolism, but also you can differentiate between oxphos and glycolysis. Due to cancer, it have more involved in the glycolysis approach. Oxphos takes place only in the mitochondria, glycolysis in the cytoplasm. This way, you can differentiate between oxphos and glycolysis by using our software approach. So here you can see the whole cell versus the mitochondria, and you can see the difference between the distributions of the signal from the mito versus the whole cell. You can able to extract information between oxphos and the glycolysis. So how do we test our method? That is the next one, where, as you know, the TC Krebs cycle, which is TCA cycle, is related to the oxphos is involved in the mitochondrial membrane, and we would like to test how the software works. So the cobalt chloride blocks the connection between Krebs cycle and oxphos. So if you add the cobalt chloride, it blocks the Krebs cycle. So the production of coenzyme is decreased, you can see. And this is the top panel is intensity based, the bottom panel is a lifetime based method. We used 50 micromolar cobalt chloride, and we added the 25 millimolar glucose and kept it for 30 minutes. And you can measure the coenzymes in the intensity ratio and then add the toxorubicin for different timing. If you compare the intensity versus the lifetime technique, you see the sensitivity, you can see the green arrow. The sensitivity increases with our technique. You can clearly see the observation, the increase in sensitivity of our lifetime technique. So here is the LINCAP cells and African American cancers, prostate cancer cell line. The response for the drug from the Caucasian prostate cancer cell is very responsive. Compared to the African American cancer cell line, the response is very less. And also you follow the coenzymes of the NADH, bound NADH. You can see the response increases with increase in toxorubicin timing. So the black color represents the control, and this is after drug treatment. The African American cancer cell line response is very diminished. And also the bound FAD also decreases, where you saw the bound NADH increases and the FAD decreases. FAD is always available in the mitochondria. So you can see the African American cancer cell line response also shows. So we also used biopsy tissue, a prostate cancer biopsy tissue, and verify the technique is reasonable for our measurements. So it clearly shows this is the lifetime-based technique on the left, on the right, intensity-based technique. It's not reproducible and also not uniform. So why we want to measure this kind of approach? We want to improve the sensitivity of the measurements. Right now we are doing on the patient samples, blood samples, testing to see whether we can able to detect the prostate cancer at early stage. As I mentioned, the PSA test is used. But through PSA test, you can see the prostate cancer. You can observe the prostate cancer only after its cancer is developed. We wanted to see whether we can detect below 4 PSA, level 4 PSA. The 10 PSA is cancer. So we wanted to see whether we can able to detect before the development of the cancer or at the early stage. So now I am going to talk about the thread between tryptophan and NADH. As you know, as I mentioned before, tryptophan also related to the cancer. Tryptophan level decreases when the cancer level increases. So tryptophan is an amino acid, and it also connected to neurodegeneration, cancer, and immune regulations. So you can able to studying that tryptophan is also provides number of pathologies related disease, age-related disease. Also, you can investigate using the tryptophan by monitoring the tryptophan level in the biological samples. So tryptophan as an essential amino acid is a precursor for niacin, which in turn is a precursor for NADH. And increased tryptophan catabolism and increased indolamine 2,3-dioxygenase activity in the kynurenine pathway are linked to cancer development and progression. There is an investigator PI in the medical school working on kynurenine pathway to investigate tryptophan involvement in cancer progression. Probing tryptophan is therefore clinically relevant. Tryptophan is clinical relevance includes pathologies in cancer and neurodegenerative diseases. Tryptophan is also used as a marker for the level of protein abundance. And we hypothesize that why the tryptophan signal is decreases. We assume that NADH interacting enzymes carrying tryptophan residues may be responsible for the quenching of the tryptophan. So if quenching tryptophan occurs, the energy transfer efficiency increases. So you can see the energy transfer efficiency, this level is the basic, the black color is a base level. You see increase in, the red color is increase in energy transfer efficiency of the Caucasian cell, and this is for African American cancer cells. So how do we prove what we are measuring the energy transfer efficiency between tryptophan and the bound NADH? So what we did is that if you use all the images we have shown is collected within few seconds, not hours. So when you leave the toxorubicin for a longer time, 12 hours, 24 hours, you get apoptosis. That means cell will die. If the cell dies, there won't be any amino acid or NADH interactions. We can see the threat. So you see that we expose the toxorubicin, say, for 24 hours, and then measured the bound NADH and the tryptophan, estimate the energy transfer efficiency. As you see, the morphology of the cell in Caucasian cell line, LINCAP cell line changed. There is no energy transfer. It's all increase in energy transfer efficiency, and African cancer cell morphology somewhat maintained, but again, there is no energy transfer efficiency. It clearly shows that the enzyme, the coenzyme carrying the residues is interacting with the tryptophan, and the tryptophan lifetime decreases. That way we can able to calculate the energy transfer efficiency. Metabolic changes of, so far I told you about the prostate cancer cells. Now I am going to show you some work we have started working on because the cancer center director is interested after showing the results on the prostate cancer, and he is interested in working on the leukemia cancer. So we started working on the leukemia cancer cells because the leukemia cancer cell will not stick on the cover cell if it is a non-adherent AML cell. So in the prostate cancer cell, you can just plate it and you can image the cells in the microscopy systems. the cell should not move when you wanted to use the microscopy imaging. You see the cell is moved. It's a distortion takes place. You cannot get anything done. So what we did is we are designing a microfluidic trapping device. And you can see this small valve, micro valve. And that is used to trap the cells. And so that we can able to investigate our time-lapse imaging with the drug. You add the drug and then with the time, you can able to record and measure the rate of metabolism. We use the acute myeloid leukemia AML cell, and the drug is ceramide nanoliposomes. So you can see that each microvalve has few cells, at least 4 or 5 cells. The green color, you can see that after adding the nanoliposome CNL, after 12 hours, you see the drug response from the cells. But if it is in more number of cells, it's not a good idea. So we would like to have a single cell so that the accuracy will increases instead of investigating all the cells together, 4 or 5 cells. So we are designing a microfluidic device so that it can trap only one single cell, so that the measurements will be more accurate of the drug response. So we are working on that. So we collect a large number of data, and working, the students spend a lot of time, a week, to process all the data with the time lapse, 60 minutes, huge volume of data. So instead of that, we are now developing a machine learning approach where you can collect the data, process the data within few minutes, maybe 2 minutes or 3 minutes, instead of working, asking the student to work for 1 week to process all the data to plot it. So right now we are working on the machine learning method where this is we give the input parameter. One thing I forgot to mention is that the FLIRR, the method we used for prostate cancer cell, we used only 2 parameters to show the increase in coenzymes with the drug. But the lifetime gives a large number of data, so about 40. So now here the machine learning, we are using 10 parameters instead of 2 to extract the rate of metabolism, how it responds. So here we are using, if you are familiar with the machine learning, we are using autoencoder and supervised learning approach to autoencoder method to extract the information. This is the input and this is the output. So you can see here, this is our 2 metric FLIRR method, shows the increase in coenzymes signal, metabolic signal, but this is the machine learning approach. You can clearly see this is used to 10 parameters. It shows a very clear sensitivity of the measurement increases, and also it is fast. You don't have to spend a week to calculate for all sets of data. You just add it and you can be able to extract the cancer cells results or metabolism. So in summary, as I showed you, the methodology we developed is more sensitivity, which is using bound NADH coenzymes and bound FAD. And this can be used for live animal and also in cells. And we have also demonstrated the interaction between the tryptophan and bound NADH. And this is the tryptophan is also decreases due to cancer progression. The evolving field of tryptophan and cancer fits well into our tryptophan research goals. So the non-invasive FLIRR assay, which we developed for the prostate cancer and machine learning, can be used to any cancer metabolic investigations in tissues and in live animals and in cells. So these are the major players: Rufon and Segupta, Zdenek, and Host. These 3-4 people are, the others are students working, and Dayan Chandra from Russell Park Cancer Institute, he provided the African American cancer cell line. and of course, funding agency. These are all previous students. And thank you.